All right, IB Bio, we are going to get our kidney and osmoregulation on. First of all, let me apologize for background noise. This is coming to you from my home. I have kids and cats and birds and laundry and stuff. So sorry, it's loud. Also, I apologize for rambling. This is probably going to be a lot longer than you want it to be. I'm going to do my best to stay on topic. So anyhow, what IB wants you to know, our essential understanding for the day, is that all animals excrete nitrogenous wastes. Nitrogenous wastes simply contain nitrogen. This is coming from the digestion of things like proteins and nucleic acids like DNA and RNA. Some animals, like us, also balance water and solute concentrations. We work pretty hard to make sure that the amount of water and solute stuff dissolved in our water is just right. IB, of course, also wants you to be always thinking about how we know stuff in science. It's a lot of curiosity. Toward the end of this, we're going to talk about desert animals and the adaptations that they have to prevent water loss. Scientists, humans, were curious about how animals could live in the desert with such little access to water, and that led to a lot of really interesting investigations and discoveries. Since we're talking about the importance of conserving water, we'll talk about why water is so important for us. So first of all, water is a really good solvent. A lot of things dissolve easily in water. That allows us to transport those dissolved things throughout our bodies, through our vascular system, our blood vessels. Water also has a really high heat of vaporization. That means it requires a lot of energy to get water to evaporate. When we sweat, a lot of energy is pulled out of our bodies to get that water to go from liquid sweat to water vapor gas that floats around away from us. That energy that is sucked out of our bodies allows us to regulate our temperature when we get too hot on the inside. Water also has a really high specific heat capacity. This means it takes a lot of energy to change the temperature of liquid water. The air around us can get a little warmer, can get a little bit cooler, and we don't change temperature on the inside too much because it takes so much energy to get water to warm up or to cool off. Of course, the outside temperature can change a lot, and then our bodies will surely follow, but it is a little slower than it would be if water didn't have such a high specific heat capacity. And then finally, super important for us, and especially for plants, water is polar. This allows it to be both cohesive and adhesive. So water can be pulled into the roots of plants, up the xylem, and out the stomata on the undersides of their leaves through the process of transpiration. Super important for plants and therefore for us, because plants are important for us. So back to animals. Animals are either osmoregulators or osmoconformers. We are osmoregulators. Things like starfish, jellyfish, sharks, amoebas, these guys are all osmoconformers. So an osmoconformer is not regulating its own salt and water balance. It's going to match whatever is in its environment. So these are all guys that live in the water. And however salty the water is, their bodies will be equally salty. So here's a jellyfish. Down here, when the environment is not very salty, the jellyfish is also not very salty. Up here, if the environment becomes very, very salty, the jellyfish will also be super salty. The jellyfish needs to be here in the middle. If the environment becomes too salty or too little salty, the jellyfish is at risk of dying. We and elephants are osmoregulators. We actually use a lot of energy to manage what our salt, sugar, water levels are. And so whether the environment is low salty or super salty, we kind of stay in the middle. We use a lot of energy to make that happen. So we are osmoregulators. So the reason that we need to be osmoregulators is because there are some pretty severe health consequences to dehydration and overhydration. So dehydration is water loss. We need to put that water back to help us maintain homeostasis. Here is a cell that has lost too much water, not enough water on the outside of the cell, too much stuff like salt and sugar on the outside of the cell. This means that the water in the cell is going to leave via osmosis, and the cell is going to shrivel up and potentially die. We don't want our cells to look like this. They are non-functional. If we have too much water, though, a bunch of water on the outside, less salt and sugar and stuff on the outside, water is going to move in. It's going to make our cells super fluffy, which is actually bad. So too much fluff is dangerous for us. Our cells can actually explode if they have too much water in them. Though before our cells explode, it's more likely that we'll die of heart failure because we won't have enough sodium on the outside of our nerve cells, on the outside of our sinoatrial node to make our heart beat, to allow our brain to process information, to tell our muscles what to do. And so we'll have some pretty severe neurological problems with overhydration. Now, dehydration, especially here in Texas, is an easy thing to suffer from. Please do not think that overhydration is an easy thing for us to catch, or to die from. The amount of water that we would have to drink in a very short amount of time in order to become overhydrated is a little ridiculous. People who have other medical problems might be more susceptible to too much water, but the great majority of us, it's not something to worry about. Dehydration is a far more common problem. Worry more about dehydration than overhydration. So back to why we have our kidneys, those nitrogenous wastes that are produced from the breakdown of nitrogen-containing compounds like proteins and nucleic acids. Those nitrogenous wastes are going to come in one of three forms: ammonia, urea, or uric acid. So ammonia is the first thing that is produced in the breakdown of proteins. It's highly toxic. It's basically Windex. We really don't want Windex floating around our bodies. So we actually turn that ammonia into urea. So our livers are pretty amazing. Use a little bit of energy, change ammonia into urea. That urea we can send to our kidneys. Our kidneys can pee it out. But if we lived in the water, we wouldn't necessarily need to process that ammonia. We could just dump it straight into the water in which we live. So fish and protists and sharks and things are actually going to just send that ammonia, that nitrogenous waste, straight out into the environment, straight into the water. It floats away in the water. It's not a problem at all. Now, if you have a fish tank, you have to make sure that you don't allow too much ammonia to build up in the water of the fish tank, because that could kill the fish. So again, we take our ammonia, our liver turns our ammonia into urea, we send it to the kidneys, and then it gets peed away. Some things are actually going to use even more energy and turn that ammonia not into urea, but into uric acid. It's super non-toxic, not a health problem at all. But what's interesting is it is non-soluble, whereas urea is soluble. So the urea is soluble, it stays in our blood, it's dissolved in our blood, our kidneys can filter it out. Non-soluble uric acid is actually going to settle out of the blood as tiny little pieces of solid stuff. This is really good if you're a chicken in an egg, and you're eating the protein that's in the egg white to grow your body. All of that ammonia that's being produced as you're a developing chicken, you don't want it floating around inside your egg, because a chicken pickled in ammonia is not going to hatch well at all. Same with urea, but uric acid is going to turn into a solid that is going to settle out of the juicy part of the inside of the egg. And then it's not a problem at all. It's just like some dusty stuff that settled at the bottom. But it takes a lot of energy. And so there needs to be a reason to head all the way to uric acid as opposed to urea. Looking at how organisms get all that stuff out of their bodies. So simpler animals like bugs have what are called Malpighian tubules. So here's the body of the bug that is using up all kinds of good protein food stuff, making some ammonia, which is turned into uric acid. That uric acid is actually put into the Malpighian tubules. So here's a close-up view here. Are all the cells of the body. They are dumping the uric acid, the bad stuff, into the Malpighian tubules. Malpighian tubules are going to carry the waste products into the hindgut, the end of the digestive system of the bug, and then they're going to dump everything into the rectum. In the rectum, good stuff like water, sodium, potassium is pulled back into the body. The uric acid stays here and then is both peed and pooped away as solid waste. Kind of amazing. We do not have Malpighian tubules. We have kidneys instead. So blood from the heart is going to flow through the dorsal aorta, dorsal for along our back, aorta, because it's going away from the heart. Blood will flow into the kidneys through the renal arteries. Renal is our word for kidney. Artery, we're still going away from the heart. So blood flows into the kidneys via the renal arteries. It is filtered through the kidney. The outer part of the kidney is called the cortex. The middle is the medulla, and then here at the center is the pelvis. The blood, after it's cleaned, is dumped back into tiny little veins that dump into this renal vein, which then is dumped into the inferior vena cava. That clean blood is sent back to the heart. The bad stuff is dumped here into the pelvis, which heads through the ureter down to the bladder, and then to the urethra, from which it is excreted from the body. So here, looking a little bit more closely at kidney structure. So again, the renal artery, blood flowing from the heart goes into the kidneys through the renal artery. Blood is filtered in the cortex and the medulla, cortex along the outside, medulla here in the middle. The cleaned blood is sent through capillaries back into the renal vein, which goes to the inferior vena cava and then back to the heart. The junk that was filtered out is collected here in the pelvis of the kidney and then is sent through the ureter, again to the bladder, and then to the urethra where it's excreted from the body. One of the things that you might be asked to do on an IB exam is to draw and label a human kidney. So here is me being an art teacher, which is never a good thing. So when I draw a kidney, a human kidney, I start with the cortex. I find it easiest to start with the cortex. This is going to be the outside, and then we're going to draw the medulla in the middle. I draw these slightly triangular chunks that are called renal pyramids. And this is the medulla in the middle. And then we're going to fill in the center with the pelvis, which is going to lead into the ureter. So there's the pelvis and the ureter. So cortex along the outside, medulla in the middle, pelvis here. My drawing would be better if I had made the medulla maybe a little bit wider, made the cortex a little skinnier. So that would actually be better. But again, cortex on the outside, medulla in the middle, pelvis at the center, pelvis dumping into the ureter. Good? Great. Kidney function. So moving away from just structure, talking about what the pieces and parts are actually doing. So the functional unit of the kidney, the part of the kidney that's actually doing the filtering, is called a nephron. You've got about 1 million nephrons in each one of your kidneys. They are tiny little 3-centimeter long collections of tubes. And they are very complex and lots of pieces and parts. You need to know them all. So again, blood coming from the heart that needs to be filtered flows into what's called the glomerulus, this ball of capillaries, through the afferent arteriole because it's a tiny little artery. Afferent is going in the efferent is exiting. So remember, E for exit. So the efferent arteriole exits the glomerulus. So afferent arteriole taking blood into the glomerulus, and then the efferent arteriole is taking blood out of the glomerulus. Now, what's happening in the glomerulus? So there are all these tiny, tiny little capillaries in this ball under super high pressure, and the blood in the capillaries is being squeezed crazy hard in the glomerulus. And the juicy part of the blood, not all of it, but a lot of the juicy part of the blood is being actually forced out of the capillaries. So the plasma, the water, the salt, the sugar, tiny little pieces of stuff, including that urea that was made in the liver from the ammonia of our protein processing, is going to, let me change colors, is going to get pulled out of the capillaries and is going to float around outside the capillaries, but inside this structure, this little cup, that's called Bowman's capsule. So again, blood is entering through the afferent arteriole into the glomerulus. In the glomerulus, stuff is getting oozed out under super high pressure. That stuff, water, salt, sugar, urea, is going to be collected in Bowman's capsule. And then that little stuff that gets oozed out is going to flow through this tube that's called the proximal convoluted tubule. Now, the big stuff in the blood, the blood cells and super big proteins and things, are too big to get squeezed out. So they're actually going to stay, and then they're going to flow through the efferent arteriole, which is going to connect up with these other capillaries known as the vasorecta. So we have all these capillaries surrounding everything, because what's happening is we squeezed out a bunch of stuff, but we squeezed out more than we needed to. And so we work really hard to pull back into the body the good stuff. So a bunch is squeezed out, and then we work hard to recover the good stuff that we want to keep. We leave in the nephron the bad stuff. So going back to our filtrate. So the stuff that gets filtered out of the blood, the urea, salt, sugar, lots of water, is going to flow through the proximal convoluted tubule. It's convoluted because it's twisty, proximal because it's close. It has close proximity to the glomerulus. So lots of stuff is getting sucked back into all of those capillaries through the proximal convoluted tubule. About 80% of the stuff that we actually want to keep is pulled back into the blood here in the proximal convoluted tubule. What's left goes through what we call the descending loop of Henle, and then the ascending loop of Henle, descending because it's going down, ascending because it's going back up. The ascending loop of Henle links up with the distal, because it's distant away from the glomerulus, the distal convoluted tubule, which then dumps into the collecting duct. The collecting duct is going to send the pee to the pelvis of the kidney, which then sends the urine to the ureter, and then to the bladder, and then, of course, to the urethra, and then out of the body. So let's look a little bit more closely at the glomerulus. So the glomerulus is in charge of what we call ultrafiltration. So it's not just filtering the blood, it's ultrafiltering the blood. So in the glomerulus, this little ball of capillaries. So here's one of those capillaries. Here's more capillary, all rolled up into a ball. So this big chunk is glomerulus. And then in the background here, you can see the wall of Bowman's capsule. So about 20% of the blood plasma, the juicy stuff, not blood cells. They're too big. But the water and salt and sugar, small molecules, about 20%, are actually going to ooze out of the capillaries and then get caught here in Bowman's capsule. How that happens, first of all, we've got crazy high blood pressure because these capillaries are crazy, crazy small. So we're tightening down the space that the blood has to be in, which is forcing the small things, the water, the salt, the sugar, out. The capillary walls here in the glomerulus are extra porous. They have more holes. We call those holes fenestrations. The way I remember this in French, fenêtre is window. So fenestrations are little windows in the capillaries that allow things to ooze out. So water, salt, sugar, tiny little things, amino acids, are being squeezed out of the capillaries through these fenestrations. In order to help maintain that high pressure, to not allow the capillaries to expand too much or even to blow up. We don't want our capillaries popping in our kidneys. That's really bad. We have these cells called podocytes. That's what these bluish greenish guys are. They're podocytes. And so they have lots of little projections that help to hold the capillaries down, but leave lots of spaces where stuff can ooze out. And again, those spaces are called fenestrations. Funny side story, my daughter's email address is defenestrationmaster. Defenestration is the act of throwing someone out a window. So there you go. So we talked about bunches of stuff gets oozed out of the capillaries in the glomerulus. The rest of the nephron's job then is to reabsorb all the good stuff that we want to keep. We basically push everything out and then work really hard to pull the good stuff back in. So here again is Bowman's capsule. Lots of stuff that's being squeezed out of the glomerulus is getting caught in Bowman's capsule, goes through the proximal convoluted tubule. And the proximal convoluted tubule, good stuff is pulled back in. About 80% of the good stuff is pulled back in here. There's active transport. We're using energy to get sodium back into the blood. So out of the tube, back into the capillaries. We have some passive transport of chloride ions because of some concentration gradients that result from the sodium transport. We also have co-transport of glucose and amino acids. These are the tiny little pieces of the proteins that we want to keep so that we can use them to build our own proteins. This is very similar to that co-transport that happens in the small intestine to get the glucose out of the small intestine and into the blood system. Then, of course, because we are moving lots of stuff out, the water will follow just because we have a hypertonic concentration on the outside of the tubule. So water will follow via osmosis, passive transport. Following the proximal convoluted tubule, again, we have the descending loop of Henle followed by the ascending loop of Henle. Here we have crazy high solute concentrations. This is super salty out here. We force a bunch of salt out. So it's super, super salty here on the outside, which of course is going to set up a hypertonic solution. Water is going to follow. And then in our distal tubule, so we leave the loop of Henle, we travel into the distal because it's far away from the glomerulus, distal convoluted tubule. Last minute little adjustments to what we have left in the urine, left in the blood. We're also going to adjust the pH of the blood. Then everything dumps into the collecting duct. Here we are going to make sure that we have just the right amount of water left in the blood as well as in the urine. So close up look at the loop of Henle because it's kind of a magical thing. So here again, we have our glomerulus, that ball of capillaries. The juicy part of the blood is squeezed out and goes through Bowman's capsule into the proximal convoluted tubule. These numbers are telling us the saltiness of the kidney on the outside of the tubes, and then the saltiness of the fluid on the inside of all of our tubes. So here we have a 300, 400, 600, 900. It's getting really salty on the inside of the loop of the Henle, the descending loop of Henle, because this part is very permeable to water. And the reason that the water is going out is because in the ascending part of the loop of Henle, we're actively transporting salt out. So we're making it super salty by pushing salt out over here. Because it's super salty down here, water is going to leave. And if we have less water, we have more salt. So here at the base of the loop of Henle is going to be the saltiest part of the filtrate, what we're turning into urine. And it's kind of weird. It's like a little bit backwards, because you would think that we would pull the salt out first and then the water would follow. In fact, it's all happening all at the same time because we have fluid in the whole thing all the time. But it is the ascending loop of Henle where we push the salt out with energy. It's the descending loop of Henle, where water, through passive transport osmosis, water is leaving the loop of Henle. So again, losing lots of water, pushing out lots of salt. We end up back here in the cortex. We have the cortex of the kidney up here. The medulla is down here. Again, medulla super salty. Far less salty up here in the cortex. And then that, what is basically urine now, is sent through the distal convoluted tubule. Here's our distal convoluted tubule, and then into the collecting duct over here. And then the collecting duct is going to send that urine to the pelvis of the kidney and then to the ureter and then urethra. So loop of Henle is all about pushing salt and pushing water so that we have just the right concentration of water going back into the blood. So all this water is getting pushed out of the urine so that it can get sucked back up by the blood. So it seems like it's bad that we're pushing water out. We make it super salty so that the water leaves the urine and goes back to the blood because we don't want to pee too much of that water away. So the length of the loop of Henle is related to the need for water conservation in animals. At the very beginning, we talked about curious humans trying to figure out how animals could live in the desert. And it's all about that loop of Henle. So a beaver that lives basically in a river or super close to a river, has access to water pretty much all the time, is going to have a fairly short loop of Henle. So here's the salty part. We only have this much time to recover water. He doesn't need to recover a lot of water. He can drink it pretty much anytime he wants to. We humans have a slightly longer loop of Henle because even though we have access to water, our ancestors maybe weren't able to live by the river all the time, weren't able to drink water whenever they felt like it. And so we worked a little bit harder. We had more space to suck more water out of our pee and put it back into our blood. Guys like kangaroo rats that are living in deserts have crazy, super long loops of Henle. So again, here's the rest of the nephron. Here's the cortex of the kidney. Here's the medulla of the kidney. All of this space is for the kangaroo rat to be able to suck more water out of the urine and put it back into the blood. That allows it to conserve more water. It's going to have super concentrated pee. There won't be a whole lot of water in his pee, but he'd rather have it in his blood than in his pee. So what if we drink a little more water, we drink a little bit less water? How does that impact how much water is left in the pee, how much water is pulled back into the body? It's all dependent on this hormone called ADH, antidiuretic hormone, also known as vasopressin, but that is not the word that IB will use. IB will use ADH, antidiuretic hormone. So ADH is made by the pituitary. Remember, the pituitary is the master gland of the endocrine system, sits here at the base of the hypothalamus. All of this is sitting kind of at the middle of the bottom of the brain. When water content is low, when we don't have enough water in the blood, the pituitary gland is going to make ADH. That ADH is going to target the wall of the collecting duct. So this whole thing, this is the collecting duct. These are cells that make up the collecting duct. Here's the middle, the lumen of the collecting duct. So this is the pee that was sent from the nephron, from the distal convoluted tubule into the collecting duct. The collecting duct, of course, is going to send it to the pelvis of the kidney and then onto the ureter. So if we have lots of water, then we don't have very many of these proteins called aquaporins. And just a little bit of water will get sucked out of the pee and put back into the blood. So here's the bloodstream that'll hold on to that water, not let it get peed away. But if water content in the blood is too low, we make ADH. That ADH is going to stimulate the production of more of these proteins. They're called aquaporins. We get more aquaporin proteins on the wall of the collecting duct. More water in the pee is pulled out of the pee and put back into the bloodstream. So more ADH means more aquaporins, means more water is pulled out of the pee in the collecting duct and put back into the blood. If, however, we have lots of water and not enough stuff dissolved in our water because we have too much water, then we have fewer aquaporins because we have less ADH, and then we're going to have less water pulled out. Any kind of antidiuretic, anti-bloating medicine is going to cause us to make more ADH. We're going to pull more water out. Reverse that. So any antidiuretics are going to help us keep water. Diuretics are going to help us pee more away. So caffeine actually is a diuretic. It is going to help with bloating. So what happens with caffeine? We don't make ADH. We have fewer aquaporins and more water is going to stay in the pee. So diuretics help us get rid of water, but be careful because sometimes you don't want to get rid of too much because then you end up dehydrated. So antidiuretics are going to pull more water into the blood. Diuretics, like anti-bloating meds, caffeine, are going to reduce how many aquaporins we have. More water will stay in the pee and we'll get rid of more water. What if we have problems with the kidney disease, kidney failure? So if our kidneys aren't able to do their job, all the bad stuff stays in the blood and basically coats all of our organs with ammonia, Windex. We don't want that to happen. That's really very bad. So we have hemodialysis here in the States. We just call it dialysis. Basically what happens is, so here's our patient whose kidneys are not working. Blood is pulled out of the patient, sent through all of these machines. Bad stuff is taken out. Good stuff is put back in. We check to make sure that the pressure is good. We put some salt back in. We make sure that we don't let any air bubbles go back into the body. And then that clean blood is pumped back into the person. Seems easy enough, but somebody who's going through dialysis is going to spend 3 to 4 hours 3 times a week, generally, to get that blood cleaned. So basically all the blood, not all at once, but slowly over the course of 3 or 4 hours, is pulled out of the body, sent through the machine, and then back into the patient's body. It works for a while. It's not a perfect process. It can generally get people through to a transplant, which is what we have going on here. So our kidneys are pretty high up in our torsos. Surgery to get to those kidneys is actually pretty intense. And so when a person is already sick, when they have damaged kidneys, putting them through a surgery where we're cutting into this part of their bodies is actually pretty rigorous. I mean, we're talking about moving ribs around and all kinds of really painful stuff that might be really hard to recover from. So someone who's already ill, we're not going to put them through the suffering of taking out the bad kidneys. We're going to leave them right where they are. Instead, we're going to add a third kidney. That's right, we're adding a third kidney down here where it's a little bit less difficult of a surgery to recover from. Now, the person who's donating the kidney, they're going through the painful surgery of breaking ribs and getting to these kidneys way up here. But they generally are healthy, and so it's easier for them to recover from that more extreme surgery. The person who gets the new kidney, it sits down here. They keep 3. It's kind of cool. Here's a quick review. Let's see if we can name all the pieces and parts of the kidney. Pause the video, try to label, and then turn it back on for answers. Are you ready for answers? Okay, good. So here, the outside of the kidney is called the cortex. Here in the middle of the kidney, this is the medulla in the middle. And then, of course, here we have the pelvis. The pelvis is going to lead into the ureter, which is going to send all that pee down to the bladder and then the urethra, and then it is excreted out. The blood that needs to be cleaned comes into the kidney through the renal artery. Renal for kidney, artery for away from the heart. Once that blood has been filtered, it is sent back to the body through the renal vein, which then leads into the inferior vena cava that sends all that clean blood back to the heart. Here, close-up view of the nephron, which is the functional unit of the kidney. So blood is flowing from that renal artery, which is going to branch into tiny little pieces. Those tiny little pieces are called arterioles. That renal arteriole is going to branch into an afferent arteriole. Afferent arteriole is going to take the blood into the glomerulus. The glomerulus is that ball of capillaries on the inside of this cup. This cup is Bowman's capsule. Bowman's capsule. So again, all of the stuff that gets squeezed out of the glomerulus is going to be captured by Bowman's capsule. The rest of the blood, so minus some water, some glucose, some salt, some amino acids, and that urea. So the blood that leaves the glomerulus through the efferent, E for exit, efferent arteriole, still going to have lots of blood cells, still going to have big proteins, but less water, less sugar, less salt, less urea. All that stuff, water, salt, sugar, urea, that's captured in Bowman's capsule is going to go through this crazy long tube known as the proximal convoluted tubule, where we have lots of reabsorption take place. The sugar that we need to keep, the salt that we need to keep, amino acids that we need to keep, these are all going to be sucked back into the blood. So we're taking it out of the pee, putting it back into the blood. That proximal convoluted tubule is going to head into the loop of Henle. The loop of Henle, where we are going to push out a bunch of salt so that we can push out a bunch of water. and then that water is pulled into the capillaries so that we can keep it in the blood. So again, here's descending loop of Henle, ascending loop of Henle. We're recovering lots of water here. And then the loop of Henle links up with the distal convoluted tubule, which then dumps into the collecting duct. And here, the collecting duct is where ADH will put either more aquaporins in so that we can pull out and keep more water, or the pituitary gland will not make ADH and will leave all that water in the pee. The collecting duct will take the pee with whatever water is left to the pelvis of the kidney right here, and then to the ureter, to the bladder, to the urethra, and out. And that, my friends, is the end of what you need to know about the kidney. I hope this wasn't too terrible. I hope you're enjoying your break. And let me know if you want to meet at a coffee shop to do something fun, like study.